Welcome to this Tim Sandle video production.
Hi, well, welcome, and thank you for joining us for this presentation. So what we're going to have a look at is clean rooms and contamination control. My name is Dr. Tim Sandle. Welcome. So in the world of pharmaceuticals, you can divide pharmaceutical products into two groups. So you have non-sterile pharmaceuticals, so this would be things like tablets and ointments and so on, and there the concern is not having certain bacteria, for example, that might be pathogenic. The other group of pharmaceuticals is sterile pharmaceuticals, and sterile pharmaceuticals are then divided into two separate groups. There are those that, as a final measure of protection, you can sterilise, so you can superheat them or bombard them with radiation. And there's another group which are aseptically filled products, which means that when the liquid goes into the container and it's sealed, you can't do anything else with it. So you can't do that extra step of prevention. So what happens to the product is that they pass through a sterile filter, they go into sterile vials, but we have to make sure that all the steps beforehand are there to reduce contamination, and all the processes involved in the actual filling of the product are designed to reduce contamination as well. So we have to have a very, very high level of focus about keeping microorganisms out and away from the actual products. Okay, so every single part of the operation in the large factory, Building 27, takes place in a clean room. And a clean room is a particular definition of a space that hits a particular level of cleanliness. And this level of cleanliness is defined by the permitted concentration of particles that are in the air. And it's particles of a particular size, which we're going to have a look at in a minute, that are of key concern. But the whole concept of the clean room is that you have clean air and clean surfaces which minimise the potential for the product to become contaminated. So there's a number of concepts which go on to make the clean room clean. So the air is filtered. We'll have a look at one of the filters in a minute. So the air passes through a HEPA filter. So that's a high efficiency particulate air filter. And HEPA filters are commonplace. So if you have a fancy vacuum cleaner, like a Dyson cleaner, that will have a HEPA filter on it. The key difference being that the HEPA filters used in the factory are of a much greater standard. There's also the concept of clean-up times. So this means that if contamination is produced in the clean room, the room is capable of removing that contamination, which is a combination of the rate that the air is coming into the room against the rate that the air is poured out of the room. And typically, a clean room cleans up, replaces its air volume in about 3 minutes. There's also the concept of pressure differentials. So the pressure differential idea is that if you have two rooms next to each other and one is intended to be cleaner than the other, then you always have the room that's supposed to be cleaner at a higher pressure to the room that it's adjacent to, which means that if you open the door, the room from the cleaner area blows out very fast and the air from the less clean area cannot physically enter the clean space. And then there's also wearing of appropriate clothing and the actions of cleaning and disinfection. And then to verify this, then the microbiome department go out throughout the factory and they're taking different samples using agar, which is the jelly-like substance, which provides a growth medium for bacteria, and by sampling the number of particles in the air, and even if you're working in the aseptic filling area, samples directly from people's suits and from their fingers. And to give you an idea of the numbers of permitted microorganisms, so the clean rooms at BPL are divided up into 4 different grades. And we'll have a look at those grades in a moment. But grade D is the lowest level of cleanliness. And there you're allowed no more than 200 microorganisms per cubic metre of air. That then reduces into the grade C to 100. And in the grade C, you'll be doing more of the final formulation of the product. In grade B and grade A, which is where aseptic filling takes place, then the levels are much tighter at 10 and then no more than 1. So you can get an idea of how microbial contamination needs to be reduced as you start working through the factory and the product gets closer to its final state. Okay, so this is a map of building 27, and each one of these squares represents a clean room. And so what we do is have the grade D clean rooms, roughly in this area, and this is where plasma is first handled. So this would be the breaking up of the plasma packs. This would be where the plasma goes through thawing, and then it goes through conditioning, and then the ethanol fractionation takes place, where you begin to get the product streams forming. Then we move into grade C areas, which are highlighted in this blue area here and over here. And in these areas, the product undergoes its formulation. So the active ingredients are added, the buffers are added, the mixing takes place, the purification. And we get to what we call the bulk product. Then the product is then filtered into here, which is the grade B area, and that filtration is sterilising filtration. The components that help with the filling area in this sterile supplies area are prepared by autoclaving or by a process called depyrogenation. And then these are all the grade B rooms inside here, and inside those are some grade A areas for the actual filling of the product, which is the highest level of cleanliness. So all told, and a number of different people count and come up with slightly different figures, but if you count the grade A areas as well, then there's about 215 different clean rooms, which makes BPL one of the largest manufacturers in the UK in relation to number of clean rooms. Okay, so summary of what I've said. So the initial part of the process, the handling of the plasma, which is less clean process, takes place at grade D. Final formulation and sterilising filtration at grade C. Handling of any components, the vials and stoppers and the like, also takes place at grade C, so they can be then put through sterilisers under clean conditions. And then the aseptic filling area is grade B, and then the filling is under grade A. And if you remember those microbial limits we looked at earlier, we're going from no more than 200 microorganisms permitted to no more than 100, again no more than 100, no more than 10, and here we're targeting less than 1 or ideally 0 microorganisms recovered. And that's all part of this protecting the product and ultimately protecting the patient. Okay, so to get an idea of microorganisms and particles, so microorganisms outnumber us on the planet by a huge factor. So if you take a gram of soil, then in that 1 gram of soil there's estimated to be 40 million microorganisms. If you take a common drop of tap water, then there could be up to 1 million bacteria in that tap water. Not harmful bacteria, but just gives you an idea of the scale and the numbers. In terms of the human body, and we're going to look at something called the human microbiome in a minute, but there are more microorganisms in our body than there are human body cells. And we actually now there's evidence that we need those microorganisms in order to keep ourselves in a healthy condition. We also carry huge amounts of microorganisms on the outer layers of our skin, and we're constantly shedding dead skin cells and hairs every day. And it's estimated that about 10% of the shed skin cells are carrying microorganisms on them. So, in an idea there, this is an idea of some of the challenges we face when we're making our products. If we were just to walk in the factory with our everyday shoes on and we just walked across the grass, for example, then you can see the sheer scale of the number of microorganisms we might be tramping into the facility. Therefore, shoe change is essential. In terms of the number of microorganisms we could pick up from different parts of our body, this is why hand sanitisation is important, wearing clean room suits is important. And the clean room suit should be thought of as a kind of a filter. What the clean room suit is doing is keeping these organisms in and stopping us excessively shedding these into the environment. So we're all about keeping those levels down. And although a lot of bacteria are not harmful, majority do some good or just not harmful at all, there are a number which are, and even that 1% could prove challenging. And one thing we'll come back to a few times is that when we look at clean room design, it's very easy to build a clean room that works really well. The problem is when you put people inside the clean room. And there's lots of research that's been done to say that most of the contamination in the clean room and most of the risk to the product comes from ourselves. So we are the biggest contributors of contamination. Okay, so give an idea of microorganisms, that they're called microorganisms because they're not visible to the human eye, and they require a microscope with at least a times 100 objective in order to actually begin to discern them. And there are different shapes. So bacteria and fungi are the key concerns within the microbial contamination control program at BPL, and they're very different organisms. They belong to different kingdoms. Bacteria are far simpler, and fungi are more complex and closer to plants than they are to bacteria. And the bacteria come in two shapes. They're either rod-shaped or they're what's called coccoidal, which is a small spherical type shape. And then the fungi are divided up into yeasts, so a common yeast like Candida, for example, or they're divided up into moulds, which give if you get black spots appearing in your bathroom, then that'll be the kind of mould that might be there. The other type of microorganism are viruses, and there's a debate about whether a virus is something that's alive or something that's dead, because it doesn't have any activity outside of a human cell, it then becomes active when it enters a human cell. And obviously viruses with the coronavirus COVID-19 is a big subject in the news at the moment. So for microorganisms to grow, they need a number of factors. And in that sense, it's not dissimilar to us. For us to thrive and be healthy, then we need a certain temperature. And the microorganisms we're most concerned about are those associated with the human body. And because they evolved and are conditioned with the human body, then they kind of like nice temperatures about 25 to 30 degrees. They also need moisture. All microorganisms to grow need a degree of water. And they also need food substances. So these are growth promoting compounds. So in terms of a pharmaceutical manufacturing environment, you can't always do very much about temperature, because the temperature might be required for operator comfort, or it might be required to condition the product. But in terms of risk factors, we always want to avoid excessive moisture, and having water spilt on a floor, for example, and not cleaned up can provide a good opportunity for microorganisms to grow. And we want to minimise the amount of available food substances. So protein, for example, is something that a lot of bacteria like. So regular cleaning and disinfection are important for bringing those numbers under a level of control. Okay, so we mentioned particles and the definition of the clean room. So what makes something grade D, C, B or A is the concentration, the number of permitted particles in the air. And the way that's assessed is by taking a sample of a certain size, which is a cubic metre. And to do that, you use a special device called a particle counter. And particle counters have lasers inside them. So they have a pump which pulls a certain volume of air in. And normally they're pulling in about a cubic foot a minute. And the air passes through a laser and that scatters. And that scattering effect enables the particle counter to say how many particles were there, and then also what size are they. And the size part is important because one, it's defined in regulations, and two, it gives a warning about the level of contamination that might be present. So just to give you an idea about the scale, if you pull a hair from your head and hold it lengthways, and you imagine that orange circle is the, you can just about see it, but we've blown it up. So the kind of particles we're looking for are around that small blue dot in there. And that size of particle is close to what the size of a bacteria might be. This kind of particle here, the yellow one in the middle, is closer to what maybe a flake of skin might be. Okay. And then going back to these grades, then what we have here is the grades going down there, and then there's two particles which are measured. The 0.5 is something that could be bacteria, might turn out to be bits of metal or bits of plastic, but there's a risk. And the 5 micron is more representative of larger skin particles. And these numbers here are the maximum number that are allowed in European law within a clean room of a given grade. So one of the things we're trying to do is to say, are those number of particles in control? Are we manufacturing and producing our products to the right level of contamination? Another thing that affects the level of particles is not just what might be there or what might be there if you turn on a piece of equipment, but it's also the way that we behave. So one of the things in a clean room, particularly for anybody working in the aseptic filling area, is always to engage in slow, deliberate movements. And you see here that, first of all, why clean room garments are important, because this is the number of particles that can just be produced through shedding. So if I was to go into a clean room wearing what I'm wearing now, then I'd be pumping out particles. But if I'm wearing a clean room garment, then that number of particles reduces quite substantially. The other thing is the way we move. So the act of moving produces more particles than the act of sitting, and the act of running produces even more particles. So we have to move around the clean room, and what we mustn't do is run around the clean room. If we run around the clean room, we'll just be shedding, and the ability of the clean room suit to contain those particles will be much reduced. Okay, so we touched on some sources of contamination, and here we're going to talk about differences between sources of contamination and then the way contamination can spread. So there's a level of contamination in the environment, there's a level of contamination from materials, and particularly things that are introduced into clean rooms. So there's a approved list of what's allowed to go into a clean room, and there's approved procedures that tell you whether that needs to undergo any kind of treatment, or whether it needs to be wiped with a disinfectant or passed through an autoclave, for example. Then there's the way things are handled. There's going to be a degree of contamination produced from machinery, but foremost, the contamination is produced from people. Okay, so another representation of contamination sources here. So again, we have people, we have water, and BPL does use a lot of water. You get the opportunity to look around the water generation plant, for example, you'll see just how big it is. And within this region of the M25, BPL is second to Heathrow Airport in the amount of water that it uses. It's huge quantities of water. Then we need to make sure that we're controlling the air, that we have clean and disinfected surfaces, and that equipment is properly maintained. And if equipment needs to undergo maintenance, that's done in a way that minimises contamination. So we may have items of equipment that are kept in good order and they're regularly cleaned and disinfected. But if something goes wrong and an engineer needs to get into the innards of the machine, it might be that opening up a panel, for example, then might add a contamination risk to an area. Okay, so we've spoken about people and the risks that we present to the product. And there's a number of ways that we can spread contamination. So we mentioned skin flakes and the level of skin shedding that takes place. And that sometimes varies between people. So we don't all shed at the same rate. And for people who are working in the aseptic filling area, then that's something that sometimes needs to be paid attention to. So it might be sometimes that more aggressive shower gels, for example, might lead to slightly more shedding. And sometimes people, where we all vary with our different bodies, that some people it might be better to shower in the morning compared to an evening, or the other way around, in order to keep contamination levels under control. The other thing is hair. So we saw that we can sometimes lose 150 hair a day. Another big one is coughing, and also the way that saliva is produced. So although the critical operations involve wearing a mask, if someone is coughing excessively, then the ability of that mask to keep contamination in is reduced, because if the mask starts to become wet, its ability to work as a filter decreases. Then clothing material. So clothing does produce fibres which spin off. So the clean room suits that are worn are relatively quite expensive as items of clothing. They're several £100s each, but they're designed to be low fibre shedding. And then the other activity is by touching. And touching is an easy one to get into the habit of spreading contamination. So if I've disinfected this surface here, but kind of absent-mindedly put my hand on the table there where there is contamination, and then go back and touch this surface, then that transmission route is quite a key one. So the kind of disciplines of spraying hands regularly or using tools to pick things up is an important one. Another thing is cosmetics, and cosmetics are a big source of contamination. They can produce thousands of particles. So to go into a clean room, it's not permitted to wear any cosmetics whatsoever. Okay. And the other thing is, in terms of control, as we mentioned earlier, anything going in, we can buy single-use, sterile, disposable technology. We can put things through autoclaves. We can apply disinfectant to things and surfaces. But with ourselves, then there's all we can do is to wrap ourselves up in appropriate clothing, wear masks, gloves and goggles, and walk slowly, deliberately, and ensure that our gloved hands are regularly sanitised. Okay, and just to give an idea about the association of the human body and microorganisms is that there's a concept called the microbiome. And the microbiome is the microorganisms and their genes that are found on or inside the human body. And this relationship between microorganisms and the human body is very complex. And there was a study that was conducted called the Human Microbiome Project, and it was run by the US National Institutes of Health. And what they were doing were taking advantage of the latest genetic techniques to not only look at types of organisms, but how closely they're related to each other. So they asked for 300 volunteers. So 300 people in the US stepped forward, and over a 5-year period, these people agreed to be poked and prodded in various places, and every place you can imagine. And a typical person, they took about 150 samples, and they kept taking those samples over a 5-year period, and they drew up this kind of interaction with the human body. So, for example, they found out that in the gut there are 10 to the power of 14 microorganisms. So that's 10 with 14 zeros, which is a huge number. They also found out that what's in the gut varies between different people and plays a different role in health and disease. So how one person reacts to a medicine might be different to somebody else, for example. Also that this entire human microbiome, because the organisms are so small, if you manage to take this microbiome out of our bodies, then it would only weigh about 200 grams. So very small quantities, but such interactions in there. So there's some fascinating things that you can read about if you're interested in just what do we contain on and in us, and how it connects with how healthy or otherwise we might be. Okay, so just some examples on relation to the outer layer of the skin. So our hair has about 1 million bacteria associated with it. The forehead has about 100,000 microorganisms. And the interesting thing is they're going to be different types of microorganisms. Okay. Then from the nose, we're into 10 million microorganisms are associated within the nose and around the nares of the nose as well. Saliva even higher, so that's 100 million bacteria. On the hand, around 100 to 1,000 maybe on the hand. And under the armpits, then again up to the level of 10 million. But you're going to get, with the human body, again this microbiome has said that the human body is made up, the outer layer is made up of different ecological niches. So because under the arms is moist and warm, and it can go up to close to the 37 degrees, you get completely different microorganisms to those that you might find on the ends of your fingers, for example. So you get different challenges, different risk factors, and where on the body we're looking. Okay, so as well as people, there are issues around with water. And water is not only a growth source for microorganisms, so microorganisms need water, so there's a whole population who will gravitate towards water, but it also allows contamination to spread. So the dripping of a tap will produce aerosols that can spread over a reasonable distance. If we're working in the manufacturing facility and there's a puddle on the floor, and you walk through it, then that activity is going to be producing aerosols as well. So water, in some senses, a double whammy, because not only does it allow organisms to grow, it's also very effective at spreading them. The other thing with water organisms is that they're amongst the fastest growing ones. So if you've heard of an organism called Pseudomonas, then this organism can double in size every 20 minutes. So if you have a few hundred cells, then within a couple of hours you could end up with a vast number of quite nasty organisms. So it's important that we don't leave puddles on the floor. So if a piece of equipment does leak or we spill water, then we move in fairly quickly and we're drying that up and then applying a disinfectant. So we really want to target less than 10 minutes, and that's because the fastest growing organisms will start growing very quickly within 20 minutes. And we also want equipment to be left dry. So some of the most common reasons for contamination in the factory is due to wet equipment. So it's equipment that's been cleaned, great, but it's not been dried properly, therefore it becomes recontaminated. It's then used for a production process and we're adding contamination into the product ultimately. And the other thing is where we use tubing to draw water from taps and so on. When we're not using that tubing, we want to ensure that we're draining it and then we're hanging it out properly so that is itself dry. Okay, the other thing is the risk of air and how the air blows in the clean room. And we mentioned earlier this concept of the HEPA filter, and the HEPA filter is controlled by a larger system called HVAC, which is heating, ventilation and air conditioning. So this system ensures that the air being presented to the clean room is filtered. So the outside air, there could be 1 million particles. The class of HEPA filter that BPL uses is called H14 class. And that standard means that for every 10,000 particles that are directed at the HEPA filter, in theory, only 3 particles can squeeze through. And that's because the HEPA filter, if you open one of these up, then it looks like lots and lots of sheets of cardboard-like paper packed into a concertina effect. But what it does, it traps particles by inception. So it stops them getting through, which means that the air entering the clean room is of a low particulate level. And then we design the clean room so that the air is in constant motion, which helps to stop particles falling out of the air and potentially contaminating. The other thing is about surface control, and this is the whole concept of touching or where we're working. And we have to assume that most surfaces, unless they've just been disinfected, are most likely to have a level of contamination on them. And it's this whole risk of transfer, which is the biggest risk. So we need to practice regular disinfection and we need to use appropriate tools. So again, working in the aseptic filling area, we would never actually touch a surface or touch a vial directly. We'd always use things like sterile forceps to do that activity. Okay, it's also important that we keep equipment cleaned, and that in the first place the equipment that's purchased is suitable for a pharmaceutical process. So there's a concept called quality by design, which means that you work out what the quality should be before you buy it. You don't buy something and then try and build quality into it. So you need to consider things like how easy is something to maintain. If I put this piece of equipment in a clean room, is it going to pump out loads of particles and ruin my environment? Do we have a system in place where we can keep clean equipment away from dirty equipment so their paths don't cross and they're not stored next to each other? And then we've got this whole wet equipment issue that we've discussed earlier, where we want to avoid that at all costs, and just this whole issue of a piece of equipment that we believe to be clean becoming recontaminated. Okay, it's also important that we're buying materials of the right quality. So this includes consumables, so it might be that the gloves, for example, we're buying are made of a particular standard. We don't want to buy gloves that if they come into contact with a cleaning solution, they can start breaking up, for example. The raw materials, so these are the extra ingredients that go into the product, are also from a suitable source. So for example, with the Gamaplex product, that requires a sugar called sorbitol, which is an example of a raw material. But that material needs to be of a low microbial level. There's no point having a very clean process and then adding microorganisms into the process. It's also important that no contaminated materials are taken in. And as materials come over from, say, warehouse, that they go through a process where layers of packaging are removed through different steps, that nothing is wet or damaged that's going in, and we never have cardboard going into the clean rooms. Of all the different types of packaging, if you compare a laminate or a plastic next to cardboard, cardboard is a big source of bacterial spores. So we always have to have, when things come in, that they go through stages of unpacking before they get to the most critical areas. Okay, so we've also spoken about sources of contamination. Now we're going to look at routes of contamination. So the sources are how could it be there, the routes are how can it spread. And this wonderful photograph here is a stop motion photograph of somebody sneezing. It's an idea of how far the projectile from the sneeze can spread. Now a lot of that will be contained if that person's wearing a mask, then 99.9% of that would be contained. Not wearing the mask and sneezing in the clean room goes a long distance. So we have to be very mindful of this human level of contamination. But going back to the air, we have this need to have the air in constant motion in most clean rooms, so it's what's called turbulent airflow. But we have to be a bit mindful about where we situate different pieces of equipment. So for example, if we have too much equipment, or let's say we're using a cabinet to do some work, if we put too much in there, we're going to disrupt the airflow, we're going to create eddies and velocities, and things might end up in the wrong place. With water, we've mentioned about the dripping tap, or the person running through the puddle, which can then spread contamination over a wide distance. And then people to people, and then there's people touching surfaces and then going to product, or surfaces going near product. So there's all these factors to take into account. And you can see here just how far some germs can travel through different human activities. Okay, so a lot of these things need to come together to develop a concept called sterility assurance. So sterility assurance is everything from the beginning of the process through to the end of the process to say, how can we put in place best practices to stop the product becoming contaminated because that product is going to a patient. So we need to be mindful of people. Training is really important. Being trained to go into a clean room, being trained how to wear gowns correctly, being trained to clean properly. The way that the rooms have been designed, how they interconnect, is it a logical flow? So if we need to go from a grade D area to a grade C area, then we go out and we maybe change our suit before we go into that area. The importance of filling the product aseptically, the importance of having good air controls from the ceilings, and there's also the importance of running media simulations. So media simulation is a way of every 6 months of putting culture media through the process to see whether we would get any microbial growth, which might indicate whether the product itself was contaminated. And then there's good disinfection and dealing with bad trends, responding to things in a timely manner. Okay, and then a key part of sterility assurance is contamination control. And contamination control is all about reducing the level of contamination throughout the process. So this point I've raised a few times, but it is important of this contamination control cascade. And then we have all those clean room concepts that we've discussed, the gowning, the techniques, the materials, and the importance of cleaning and disinfection. So this was the concept of the clean room. So this was the point we made about the HEPA filter. So we get the thousands of particles, which are then reduced down as the air passes through. And then we get the good mixing of the air. So just to summarise those concepts, we have the clean room. So imagine this is a clean room. The air coming into the clean room passes through the HEPA filter, which gets rid of most of the particles. If we then locked the door, we'd end up with a very low particle area. But we go into the clean room and we're shedding a level of contamination. So we want to make sure that that air is in constant motion. If the air is in constant motion, then it helps to prevent the contaminants from settling out. But after a period of time, there's a risk of that building up. So what we want to do is to have the room air volume to keep changing. So we get this concept of air changes. So it means that, say, the air in here probably refreshes itself maybe every 10 minutes or so. In the clean room, it's every 3 minutes. So the air is constantly poured out, and then we maintain good pressure differentials. The other important concept is how we look. So this is the clean room gown, and this is an example of what someone would be wearing if they were going into the aseptic filling suite. So they would go through a change process where they would have removed their outdoor clothing, and then they're wearing this special suit that's designed to allow the operator to breathe properly, but is keeping the contamination in. It's like one big filtration unit. There are goggles to ensure that there are no areas of exposed skin, and the mask, and the hat, the special gloves, and the boots, and that is a full aseptic gown process. Okay, so when you're going into a clean room, there are a number of important documents that need to be read. So there is PDN 14, which is about hygiene, and then there is PDN 295, which captures other elements of the change process. So it's very important that nobody goes into a clean room if they're feeling unwell. And by unwell, that means no coughs or colds. So if you have a cough or a cold, then the ability of the mask to contain contamination will decrease. So we know that for a healthy person, you can wear a mask for 4 hours. If you have a cold, then the ability for that mask will drop down to about half an hour. So it's just not any good because of the amount of moisture that you're producing. If you have a higher level of eczema, you start getting flaking skin, and that's going to increase the level of skin shedding, and the ability of the gown to capture that is going to be reduced. Open wounds will naturally seep, and you get a layer of moisture on the suit, which will then again let contamination out. So any of those conditions always have to be reported to the local manager and noted. There's also the concept of, you come into work for a pharmaceutical company, you have a responsibility to practice good hygiene yourself. So it does mean taking daily showers or baths, and keeping yourself in good order and reporting if you're not. Also stands that there are no cosmetics allowed to be worn in a clean room whatsoever. So if your job is working in the clean room every day, then unfortunately when you come to work you can't wear any. If you occasionally have to go to a clean room, then you need to bring in the right levels of makeup remover and so on, and be prepared to remove that makeup. There's also no jewellery permitted apart from the plain wedding band. And we did have an incident only a couple of weeks ago when somebody wore a ring into a clean room that had a stone in it, and that tore the glove very quickly, and then the operator's exposed skin was there, and that presented a risk to the product. Also, no watches. There's no personal phones, because you may have seen some of those programs on television where they say, where are the most bacteria? And then you have a quiz, and it's always, the phone often has more bacteria on it than a toilet. You're also not allowed to take in any medications, because we are a GMP facility, and it would be horrendous if even like a tiny bit of paracetamol was to end up in the product, for example. So you're not allowed to take any medicines into the manufacturing area. No chewing gum, and no smoking materials. Another thing is, again, with mask control, if you are a smoker, then you really need to have had your last cigarette about 20 minutes before you go into the clean room. And that's because studies have shown that smokers produce a much higher level of particles, and again, the ability of that mask becomes decreased. Wearing the right clothing, always have to clean your hands before you go into the clean room, and then there's various stages within the clean room. You need to keep on practicing good hand sanitisation. Wearing the right footwear, and the shoes, because remember that thing I said earlier about the number of bacteria on a gram of soil, need to be captive to the manufacturing facility. So your shoes never leave the building, and then they're subject to regular disinfection. And everybody has to have an annual health check, or a 6-monthly health check if you're going into the filling suite. And there's either an annual or 6-monthly gowning test, depending on whether you're working in grade C or D areas or working in the aseptic filling suite. So this gives a level of contamination control on us, the biggest sources of contamination. Okay, and there's just a example slide of the things that you're not allowed to take in or wear within the clean. Okay, so gowning is again a fine art, and it is a little bit of an alien concept, particularly if you're working in the aseptic filling suite. So practicing gowning is really important. So as new joiners to BPL, any opportunity you can get to practice gowning, the better. And building into for both areas, for C and D and for the aseptic filling suite, you need to show to a trainer that you can get gowned correctly. So there's a lot of key aspects to that about getting gowned in an aseptic way. So it includes that things like the suit is not allowed to touch the floor, which means that you are getting gowned, so you undo your suit and it's wrapped in a particular way, but you need to do it in such a way that you're putting your legs up and you're sliding the suit on and so on. So keep on practicing gowning is the best advice. And one reason to do that is, one, you don't want to add any extra contamination to the suit, and two, you want to minimise, if you're getting gowned in a haphazard way and it's quite a frenetic activity, then you don't want to be shedding any more and adding to that contamination risk. So practicing gowning is very important. Okay, so the other thing is going into changing rooms, and there's a kind of a cascade of that. So when you first go into building 27, there's a first stage changing room, and here you need to remove your outdoor clothing, and then you go into the areas where you'll be putting on clean room suits. And once you're in the official changing room, then that changing room is of the same cleanliness level as the area which it leads into. So those levels of microorganisms need to be controlled in the same way. So there's a difference between a grade D changing room and a grade C changing room and a grade B changing room. So there's a very proceduralised process, and it's very important that process is followed at all times. We've covered the material and the shoes. The other thing is, you may see these types of mats around, and that's a special mat called Dicen flooring. So it's made of a material, a polymeric material, and it has a static charge on it. And what that does, it takes off contamination from footwear with a charge effect. So it reduces what you can actually walk into the clean room area. Okay, and to do that, 5 steps is ideal, and 5 steps has been shown to reduce contamination by 80%, according to the manufacturer, and the manufacturer got that from a study that was done at BPL, so BPL actually did the initial verification of disulfide. Okay, so other good gowning practices. So the mask has to fully cover the nose and the mouth completely. Safety glasses need to be sanitised prior to use, and if you're going into the aseptic filling area, then the goggles have to be sterile, and you must cover the eyes. If you're wearing a hood, it has to be tucked into the gown, and it's got little press studs, it needs to be done up. Gloves, so in areas where you're required to wear gloves, it's good practice every 10 minutes to undergo a hand sanitisation exercise. And the way of doing that, which I think there's a demonstration coming up shortly, is for at least 1 minute. Okay, which is again a process that's been proven at BPL. So for that we've got a lot of volunteers, got them to put gloves on, they dunk their hands into a bacterial soup, which is great fun, and then they had to then sanitise their hands and noted how much was destroyed. The gown we mentioned about putting on properly, the careful shoes, and also where there are step-overs, the important part there is to ensure that that's disinfected before you step over or use that. Okay, the other thing are other good behaviours within the clean room environment. It is highly disciplined. Okay, so things need to be kept in order, neat and clean, and always have the bare minimum. Every extra object in the clean room is a potential source of contamination. You also want to avoid unnecessary talking. You want to refrain from shouting, because again that will affect the ability of the mask. And if you're working in the aseptic area particularly, then you have to get used to kind of standing like this a lot of the time, or sometimes like that. But you mustn't do activities that are going to result in skin rubbing or making the body warmer, so not standing folding the arms, not leaning up against objects, and so on. So it is an interesting discipline. Avoid coughing and sneezing, so if you do start coughing and sneezing, you need to exit, at least change your mask. It might just be a little bit hay fever or something, but if it turns out to be a cold, then stay away. Slow and deliberate movements. The other thing, if you drop something onto the floor, then you need to leave it to the end of the shift or the end of the session. Most of the contamination will have settled onto the floor. So it goes on the floor, just leave it until the end of the time. You should avoid always open doors using hands, or where there's automated doors with the automatic controls, but don't press up against the doors. And then for those who have to take samples, then there's a number of good aseptic practices to take and to follow when taking samples. So avoid putting your hand across samples or your finger into a sample, for example. Okay, so gloves. Now, gloves do protect the product. Okay, so they're better than bare hands, but gloves are not the solution to everything. And it's often quite amusing if you go to a canteen, for example, and you see someone put gloves on, and they suddenly think they're invulnerable to anything, so they kind of go and touch one thing, then touch another thing. Gloves don't work like that. That they do reduce the contamination on the skin, but they're not perfect. So you do need to sanitize the gloves regularly. And for glove sanitisation, we use an alcohol, which is 70% isopropyl alcohol, or IPA. Unfortunately, it's not a reference to a beer, but it's a reference to a particular type of alcohol. And it's been shown that, strangely, that 70% alcohol is more effective at killing microorganisms than, say, 90%. The reason for that is to kill the microorganism, they've got to open up their pores. And if something's too concentrated, they'll remain locked. If they think it's water, they'll open up. So their sensing is that this is something we want to take in. They take it in, and then the alcohol then kills them. More concentrated alcohol, they wouldn't actually be killed, and lower concentrations, they wouldn't either. So that's why it's 70% for the IPA. It's also important that gloves are integral. So if a glove becomes damaged or torn, then it gets replaced, a new pair of gloves is put on. And it's always important to wear gloves, anything relating to product or clean room. Okay, so for things going into the clean room, we've covered a number of these aspects, but it's always important to be correctly gowned as well to avoid extra contamination. Always avoid cardboard. Only allow authorised materials to go in, and everything going in is disinfected. Okay, so I mentioned at the beginning about the differences between detergents and disinfectants. So a detergent is something that cleans. So some detergents go under the name of surfactant. Surfactant is surface active agent, and what that is it works at the dirt and it disassociates the dirt from the surface. So what you want to do before you disinfect something is have all the dirt taken off. So if you're using a washing machine, then you're adding in the detergent. What you're trying to do is get the dirt away from the clothes, and it's flushed away through the activity of the washing machine. It's exactly the same when you're trying to clean the surface of BPL. You're trying to get that dirt to stop being stuck to the surface. And there's two different types of detergent used, one called Hospec and one called Clareside, and they're roughly around a neutral pH. A disinfectant kills microorganisms. So a disinfectant is different to a detergent. The reason you need to use both is that disinfectants are not very good at cleaning. So you need to clean to get the dirt out, and then you come along with disinfectant, which then kills the microorganisms. And there are three main disinfectants. So I've already mentioned IPA. 70% IPA is used on gloved hands. There is another hand disinfectant called Spirigel, and this is suitable to use on bare hands. So if you need to disinfect your hands when you're going for a changing room, use Spirigel. And Spirigel is denatured ethanol. And the reason for the difference there is it's been formulated not to hurt your skin. Whereas if you use the IPA on your skin, then your skin will start drying out. So you have these two slightly different hand disinfectants. And then there are two other disinfectants that are used basically for all the surfaces. And those disinfectants are rotated. So in a lot of areas it's 1 week of one week of the other. And the reason they're rotated is to increase the spectrum of kill, so you can start targeting different types of microorganisms and avoid one type lasting for longer. Okay, so what is cleaned? All surfaces, floors and walls, and these happen at different frequencies. So it depends on the grade of the clean room. So remember the A, B, C, D. The A's and B's get cleaned and disinfected more often than the C's and D's. And then all working surfaces are done before every operation. Floors often tend to be done weekly, walls at lower frequencies. And there's also a particular system as well called the triple bucket system. And the reason it's triple bucket is it's to avoid recontaminating the mop. So the mop will go into the solution, but then it needs to be wringed out and then rinsed before it goes back into the next solution to use for the next stroke. It's also important to follow the right procedures. So you always have to use detergent first, and you always need to make sure the detergent's dry first before the disinfectant is applied. You make sure that it's within its expiry date and it's been made up correctly. So there's no value in doubling the concentration, for example. Okay? You've always got to make these things up correctly. And then you've got to use the correct technique as well. So what we don't want to do, as you might want to, let's say we're cleaning our windows at home, we don't be doing this. We need to be doing even strokes, and where the second stroke just slightly passes over the edge of the next one. So we're doing these parallel strokes. And doing things like, if we're going to clean this room and the door's over there, then I always start here and I'll work my way out of the room. But I'll start over there, come back here, and then walk across the area I've just been cleaned. So there's a few things to observe there. There's also the importance of contact time. So all of the disinfectants have been experimentally tested to say that this one might need 5 minutes to kill bacteria. So if you're going to apply it, you need to wait 5 minutes, and if it starts drying out, you might need to do a new application of that. Another area of concern sometimes happens is when you're moving, say, trolleys or carts from one room to another. One of the big sources of contamination are the wheels. You need to make sure those wheels are correctly cleaned and disinfected. Okay, and there's some examples of the cleaning strokes that we discussed. So it's about the overlapping strokes and always moving in the same direction. And with a wall, for example, that's always from the top of the wall downwards. Okay, so again, the number of things that I've mentioned, you might say, well, why are we doing all of this? Well, we're doing it for a number of reasons. One is that inspected on a regular basis by medicines inspectors. If the medicine inspectors don't like what they see, then they have the power to suspend operations. We also seen that contamination poses a risk to product quality and to safety. There are challenges around toxins as well. So some microorganisms might die, but they produce poisons. There's also aspects leading into how effective our training is, how the facilities are managed, and so on. We may not be able to release our batches on time because every contamination event requires investigation. And even if it's not a high risk, we've still got to have a look at that and do an assessment. And we may be wasting time and resources and overall costs if we don't do things properly. And some of the products that we make are very, very expensive. So the Gamaplex product, for example, can often retail between £3 and £4 million for a batch of Gamaplex. There's things around workload, missing targets, and unhappy managers, but foremost is always patient safety. And it should never be forgotten that the people who need our products are people who are not very well. So they're people who already have very weak immune systems, and they need the medicines to make them well. And what we don't want to do is to present their immune systems with contamination that then makes them iller than they were before they had the product. Okay, so just to sum up, key reminders are follow procedures. Procedures, there's lots of documentation at BPL, there's lots of procedures to follow, but those procedures are based on best practice. They're designed to protect the product and the patient. Okay, please don't pass up on any of the contamination techniques. Always follow what we've done. Wear the gowns, masks, gloves, equipment, head covers correctly. Always move and work in the same way. Always follow good techniques to avoid cross-contamination. Report any concerns, so always flag things with your manager, and do what you can, because everything we can contribute as individuals helps to protect the patient. Okay, does anyone have any questions they'd like to ask?
Thank you for watching this video, created and presented by Tim Sandle. Please subscribe to this YouTube channel.